Today we're diving into the fascinating world of osmosis. And I'll explain to you the ins and outs of the process, and I'll also tell you about tonicity. These concepts are super important in biology since osmosis always happens in our cells. This is fundamental knowledge you must know, especially when understanding how cells interact with their environment. So let's get on with it. Okay, before I dive into osmosis, let me give you some background information to help you steer away from confusion. You may have heard of diffusion, but what exactly is it? Diffusion refers to the passive movement of molecules from high concentration. As you can see here, they're very close together, bundled up. So it's a passive movement for molecules from high concentration to low concentration. Molecules like to spread out and like to maintain stability, and the key word here is passive. So it is passively moving. And let me show you an example here with some water and protein milk. So here's a little cup of water, protein milk. You can see how quickly it diffuses into one. The molecules from the protein milk, they mix around and spread out quickly in the water solution. Okay, and so that's diffusion. And now, diffusion can happen across many types of molecules, but osmosis focuses on the movement of water across a selectively permeable membrane, which is represented by this line here. The membrane is less permeable towards the solute than it is to water, meaning that water passes more easily through this membrane than the solute. The solute in this example is sodium chloride, aka salt. Now, water loves solutes. Water molecules will always go from an environment from low solute concentration to an environment with high solute concentration. Now, equilibrium is reached once the concentration is equal on both sides. So over here I will show you an example. So here we can say there is a few water molecules spread around, and this is before the osmosis process. And then in order to reach equilibrium, what happens is that the water molecules that were on the left side with less solutes, which is represented by the pink circles, they like to move towards the side with more solutes. So here, I'll draw it. And then that is how equilibrium is reached in osmosis. As you can see, it went from the low concentration to the high concentration. And when learning about osmosis, it is also crucial to understand tonicity and the three types that you'll come across. And tonicity refers to the gradient in which water molecules are moving. So there's hypotonic, isotonic, and hypertonic. So in tonicity, it's important to specify what you're referring to when you're mentioning the type. In this example, I'm representing the tonicity of the solution. Therefore, the inside of the cell would represent the opposite if the environments are not equal. In a hypotonic solution, there will be fewer solutes in the solution, meaning around the cell, than inside of the cell. Since water loves solutes, it'll move inside the cell to be with the solutes. So we can say these blue circles are water molecules, and then there would be more solutes inside of the cell than there is outside. Draw one more here. And then in an isotonic solution, the solutes are more or less equal. So there's a constant cycle of water molecules passing in and out the cell's membrane. So to represent that, it would be like this. So it's more or less equal. And then in the hypertonic solutions are full of solids and are pretty much the opposite of hypotonic solutions, where the water molecules are now moving out of the cell to be with the higher concentration of solutes in the outside of environments. So in this example, it would be the solution. So yeah, so hypotonic, the water goes inside to be with the higher concentration of solutes inside the cell. Isotonic, it is more or less equal. And then hypertonic, the water molecules leave the cell to be with the higher concentration of solutes that are outside of the cell. Now, I'll let my amazing group mates take the lead here to help you understand osmosis by providing more specific examples of how osmosis can be found in biology. Thank you.
Hi, now we're going to dive a little deeper into biology and discuss the tonicity of red blood cells. In hypertonic solutions, the cell shrinks and shrivels because of the high solute concentration inside of the cell versus the low solute concentration outside of the cell. So water moves from low solute to high solute, making water move out of the cell, which ultimately makes it shrink. So what you need to know about hypertonic is that water moves out of the cell, that's why it shrinks. In isotonic solutions, there is equal net movement in and out of the cell. So the water comes in and goes out in a net movement, and it's equal, so the cell remains normal. In hypotonic solutions, there's low solutes inside of the cell and high solutes outside of the cell. So water moves into the cell, making it swell and burst. So what you need to know about hypotonic is that water moves in, and that's why the cells burst and swell. I'm going to discuss the tonicity of plant cells. In hypertonic solutions, water moves out of the cell because of its solutes, which is also known as hypertonic. Like stated before in the red blood cells, that hypertonic makes it shrink, but in plant cells, we call it a different name called plasmolyzed. So water moves into the cell because water moves from low solute to high solute. In hypotonic solutions, there's equal net movement in and out of the cell, and we call this flaccid. In hypotonic solutions, there's low solutes inside of the cell and high solutes outside of the cell. And the more water the plant cell has, the better, unlike for human blood cells. So turgid is considered normal in plant cells, and because of their cell wall.